Hey class, happy Wednesday. I am back in the classroom. Oh, all empty and sad without all of you. Pretty boring place. When I walked in here, it's been a month since I've been in my classroom. I've been home, but I have had this, my fish have been weighing heavy on me. My fish tank has still been in this classroom, and I wanted to make sure my fish were okay, and I was really worried they'd all be dead. But here's the fish tank, and yes, there is not a lot of water. I need to change that, but can you see them? They're still alive. If you look on the left, you see that minnow? It's swimming. If I go here, they're still moving. They're still swimming. They're still alive. All of them. I don't see any dead fish. So, our classroom ecosystem is still alive, even though some of you have put lunch meat in there and you've poured half a bottle of fish flakes. Those fish are happy. They look good. They look bright. They look colorful. So things are good in room 664. But this video is not about fish. It is about evolutionary relationships. And this week, the new idea is phylogeny. How do we represent evolutionary relationships? How do we organize all of life in one giant tree of life? Everything from bacteria, prokaryotes, to eukaryotes like us, plants, fungi, animals, single-celled organisms. How are they all represented from an evolutionary perspective? Now, our two objectives this week is to first visualize those relationships using a cladogram. How do we make it? How do we see those relationships using a tool, a graphic called a cladogram? And then how do we analyze evolutionary relationships using those cladograms? All right, and I just want to say for the videos I make this week, go Bears, go my alma mater. Thank you to evolution.berkeley.edu. I used their graphics and their images to help us read and understand cladogram and phylogeny. Well, for vocab, phylogeny means an evolutionary relationship between species. So when we consider Darwin's finches, those finches that have speciated on different islands due to natural selection, we can represent their evolutionary relationships on a phylogenetic tree or a cladogram. And we can, how do we organize those phylogenies? One, we can use anatomical and physiological comparisons, or DNA and protein sequences. So, using Darwin's finches, we organize their evolutionary relationship based off of anatomical features, homologous structures, beak size. Okay, we can also look at DNA sequences and organize their evolutionary relationship based off of which finches have more similarities in DNA sequences. Okay, and likewise with horses, going back in time, we can organize the phylogeny of a horse based off of its anatomical features, based off of what we found in the fossil record, and creating relationships that make the most sense. All right. Now, the cladogram is the diagram that shows the phylogeny, that shows the evolutionary relationship. So here's a cladogram of many different animals, fish, amphibians, reptiles, primates, and birds. Okay, and so we're going to, in this video, we're going to analyze this and see how we actually read it. All right. Now, the first two parts to look at are the roots and the tips of the cladogram. So the base of a cladogram, the root, represents ancestors. And the further down you go in a cladogram, the older you go in time. So in this generic cladogram, this root here is an ancestral species. And as you go up in time. And when you get to the tips of the cladogram, this is current time. This is 2020. These are descendants of ancestors and the most recent organisms. And when we read this cladogram, okay, so down here at the root is an ancestor of all of these organisms, and that through time, as we go up, the up in time, we get to the tips of the cladograms, and these are our most recent organisms. And everything in between, this is all time, these are populations of organisms that have been changing and evolving over time. Okay, and another part are these branching points. So wherever it branches, that's a speciation event. That means that a population has turned into two populations that can no longer reproduce, and they are species. So geographic isolation, reproductive isolation happened, and two species diverged from each other. And the lines going to a branch, this is an ancestral population. So one population, and through time it changed, and then that population split and changed so much that now those two separate populations can't reproduce, they can't mate with each other, and we create a branch because the speciation has happened. So when we look at this cladogram, there was an ancestral species here that had a backbone, but there were two populations that slowly began separating. Mutations in those separate populations built up. They were isolated, natural selection in different environments, started selecting for different variations, and they became so different that they speciated. And that one population formed what we know of as sharks, and another population created all the other organisms that we have today. So at each little branching point, that represents that there was an ancestral population that split and could no longer mate and reproduce with each other, and created, and that now became an ancestral population for other species. Now, each line represents a history. So it's not just the root and the tip that matters. Okay, the lines are showing time. So along this line, you have a population of organism, and that population is living, surviving, reproducing. Okay, natural selection is acting on its traits. Mutations are building up. Okay, gene flow is happening. Chromosomes might get duplicated. All these different events are happening as time goes on. Now, between a branching point and a tip, that's a unique history. So for A, B, and C, the A, its unique history is in black, B, its unique history is in green, and C, its unique history. So that means that along this line there was a population that was just that species, just species B. And that as time went on, it's been living life, reproducing, undergoing natural selection, mutating. But these dashed line, so this whole line is a shared history between B and C. So this population has connection to both B and C. So this population, as it's reproducing, undergoing natural selection, mutating, its history is shared with B and C. So if I look, so this whole long line with sharks, that's a unique history for sharks. So this population, so these population of organisms, ancestors of sharks, changing, undergoing natural selection, mutating, leading to sharks. But if I look here, this part is a shared history with all life. And so crocodiles and birds, so this little part, that is a unique history for a crocodile. This is a unique history for birds. But this is a shared history with both crocodiles and birds. Okay. And likewise with the unique history, like any point along this line is a unique ancestor, but this point going down is a common ancestor of B and C. So when I go back to this, so if I look here with primates and rodents, if I just went back in time to this point, this population, it's not a modern day primate, it's an ancestral primate, okay? It would be a unique ancestor just to primates. But if I stopped time and I traveled to this point, okay, whatever population is here, it's a shared ancestor of rodents and primates. Okay, now with clades, a clade is a grouping, an evolutionary grouping that has the common ancestor in all its descendants. So you find a branching point and you circle everything that connects to that branching point. Okay, and it could be as big as the whole entire cladogram, could be just a chunk of it or just a single line. But these are not clades. Okay, this is not a clade because these three don't share the same common ancestor. This line, its common ancestor is here, where these two common ancestors over here. Not a clade. Here, clade, because all three of these species share a common ancestor. All right, and these. So even though all of these 4 that are circled share a common ancestor, it's not a clade because it's not including all descendants. I'm missing this descendant. So I would have to expand this to include this species to make that a clade. Now if I go here, so some clade examples, the crocodiles and birds are considered a clade because I have a common ancestor. It was some ancestor that had an egg with a shell, and I have all the descendants. Okay, amphibians, primates, rodents, not a clade because even though I come down here, they share a common ancestor, I don't include crocodiles and birds. I'm not including all the descendants. Okay, or if I wanted to look, these primates and rodents share a common ancestor that is not shared with an amphibian. Okay, not a clade. Now, when we decide what's a clade, how to split things, we look at homologous structures and anatomy, physiology, and DNA. Okay, and the shared derived characteristic, this is some sort of characteristic that evolved in a common ancestor and is shared by all in the clade. And it may be unique, it may not be unique. But let's look. So in this cladogram, the tetrapod, its shared derived characteristic is 4 limbs. Now at this point, 4 limbs evolved and it is shared by all tetrapods. Kind of like in the evidence for evolution worksheet where we looked at limbs. Okay, those limbs are shared derived characteristic, and that the common ancestor had a form of that shared derived characteristic, and it's been modified over time. But sharks and fish are included in this clade because they do not have the same shared derived characteristic. So now let's look at this cladogram. So vertebra is a shared derived characteristic for all these organisms because all these organisms have a vertebra. Okay, hair is a shared derived characteristic for primates and rodents because they have them, whereas the others do not. Okay, and then we'll do eggs with shells. That's a shared derived characteristic for crocodiles and birds because they are the 2 organisms that have it. Their populations are the populations that contain that shared derived characteristic, and it evolved one time in a past ancestor. All right, so another example. Let's look. So here's our cladogram. Okay, so prosimians and humans, we could call them a clade. Their shared derived characteristic is hair. So the hair separates them from the other organisms. Okay, so if I'm thinking about roots and tips, okay, so this common ancestor, this is the oldest in evolutionary history, and as we go up in time, we have modern day organisms at the tips. So with insects, this line is a unique history for an insect. Okay, and so at the very beginning of time, there's an organism, a population, okay, that had a blastula, okay, cells that are developed a certain way, and at some point, 2 populations split. Okay, and one population changed into what we know as an insect, and another population over time changed. And so in this population, one shared derived characteristic it has is a vertebra. So this population did not evolve the vertebra, but this one did. Okay, and then time goes on. There's a shared ancestor here. Okay, and one population split off, still having the vertebra, but it did not have the shared derived characteristic of a tetrapod. Okay, and then time is going on. 2 populations again from a common ancestor split, and one changes into an amphibian, and another changes and develops an amniotic egg, a second shared derived characteristic. And then you have a shared ancestor, and then 2 populations again split. Okay, and one population forms what we know today as birds, and another population has a shared derived characteristic of hair. Okay, and then that population splits, speciation happens. There's a shared ancestor, but 2 populations speciated, forming prosimians, and then one became bipedal, forming humans. Okay, hopefully this helps with helping you understand the taxonomy practice worksheet and understand your reading for 17.2. Stay tuned for another video on how to actually make your own cladogram. Bye, class.